Okay, welcome to Roasted Academy. Today, let's discuss biochemistry exam strategy preparation. So, at least before going for your exams, you would have known these topics, right? In detail. Bioenergetics, glycolysis. So, just after glycolysis, you do 3 bisphosphoglycerate shunt, right? Um, this is very important because it takes place and very active in the red blood cells, right? And facilitates the oxygen delivery to the tissues. Also, go to the deficiencies, hexokinase and pyruvate kinase deficiency, citric acid cycle, citric acid cycle, where we have converted pyruvate to acetyl CoA that has been produced from glycolysis. Now, the acetyl CoA we channel it to the citric acid cycle, right? So, in the citric acid cycle, acetyl CoA is going to be oxidized to carbon dioxide, right? And we're going to produce those molecules. Supposed to know about each of those steps of the citric acid cycle. Pyruvate dehydrogenase complex. So under pyruvate dehydrogenase complex, you are supposed to know about the conversion of pyruvate to acetyl CoA and the cofactors that are needed for this enzyme, as well as the names of the enzyme. You know, a complex is comprised of uh, a lot of enzymes, right? So in this case, for PDH complex, there are three of them: E1, E2, E3. Okay. Electron transport chain, very much common MCQs, right? As well. You're supposed to know about all those complexes: complex 1, complex 2, complex 3, complex 4, as well as the ATP synthase. And also uncouplers also. What are uncouplers? Okay, and how the electrons are going to move from complex 1 all the way up to meeting oxygen in the end, so that oxygen gets reduced to water, then we form energy. Okay, and then gluconeogenesis, which we mostly say it's the reverse of glycolysis, but it's not, right? Because there are those enzymes that are involved which are totally different from glycolysis, right? So gluconeogenesis is simply where we form glucose from non-glucose containing molecules or compounds. Okay, like for instance, we are moving from um, pyruvate to form glucose. So at least know when do these particular pathways occur. Is it in fed state? Is it when someone has run out of glucose, right? So fed state simply means that someone is having the carbohydrates, or they have eaten, right? Not only carbohydrates. But for fasting state, that means there is no carbohydrates, or they haven't eaten, they've fasted for a period of time, right? Then know about pentose phosphate pathway. I've highlighted the oxidative phases here. Pentose phosphate pathway has got two phases: the oxidative phases as well as the non-oxidative phases. You must at least have known about the oxidative phases, where we talk about generation of NADPH, very important molecule, okay, which is used as the reductive biosynthetic molecule, right? So you can also know about the non-oxidative phases, right? And also that in pentose phosphate pathway, the generation of ribose 5-phosphate, which is used as the part of DNA, okay? Ribose 5-phosphate. From there, there's glucose 6-phosphate dehydrogenase deficiency. and its result of anemia. Fatty acid breakdown and calculations. Supposed to know about the beta oxidation of fatty acids, as well as how to calculate the total number of ATPs. Okay. Ketogenesis. So ketogenesis is the formation of ketones. There are three ketones that we talk about. There's beta hydroxybutyrate, there's acetone, as well as acetoacetate. These are produced in prolonged starvation state. Supposed to know about ketogenesis pathway as well, right? Diabetes. What is diabetes? And also the effects of insulin on carbohydrate metabolism. In fact, in carbohydrate metabolism, on protein, as well as on lipids, right? So there are two types of diabetes. There's the diabetes type 1 and type 2, as well as there's also gestational type of diabetes. But for us, we stick to two types of diabetes. Okay. Type 1, it's a insulin dependent type of diabetes. Type 2 is where there's a insulin insensitivity, right? Then fatty acid synthesis. Know about the fatty acid synthesis, how to explain all those reactions, and also that fatty acid synthase, it's a an enzyme that comprises of those enzymes that we mentioned, about seven of at least 11 of them, right? 7 or 11. Know about the fatty acid synthase complex. Okay. Then classification of amino acids. What are amino acids and how do you classify them? There are essential amino acids, there's non-essential. There are different types of classification of amino acids basically, but at least you're supposed to know about essential and the non-essential. What are essential and what are non-essential type of amino acids as well. Cysteine synthesis of tyrosine from phenylalanine, and also the disorders that are associated with these particular pathways here. Okay. Branched chain amino acids and maple syrup or urine disease. Okay. Supposed to know about that. Urea cycle. Know how to explain the urea cycle and the stages, the steps of urea cycle, as well as take note of CPS. Okay. Carbonyl phosphate synthetase 1, which is the first enzyme that is involved in the urea cycle. Right? And I've highlighted here disorders of amino acid metabolism like phenylketonuria, tyrosinemia, homocystinuria, non-ketotic hyperglycemia, maple syrup urine disease. Know everything about these enzyme deficiencies, right? If the enzymes are defective in this case. Heme synthesis and regulation. What is heme? Okay. And how do we synthesize heme? How do we break down heme? That's heme catabolism. Heme catabolism, it leads to bilirubin in the end. Okay. There's bilirubin production, and that bilirubin will simply go and accumulate in, for instance, under the skin, in the sclera. That's jaundice, right? So know about the types of jaundice, and we have got pre-hepatic jaundice, hepatic jaundice, and post-hepatic jaundice. Others can say hemolytic type of jaundice, hepatic jaundice, and obstructive jaundice, right? Yeah. So know how to explain all that. Replication of DNA here. This is nucleic acid metabolism here. So here we talk about when DNA is trying to replicate itself. So you know about DNA replication and the enzymes. There's helicase enzyme, there's topoisomerase, there's ligase, there's RNA DNA polymerase to come in, and the like, those enzymes and their functions, right? Also, when we're moving from DNA to RNA, we talk about transcription, right? Yeah. And stages of protein synthesis. How are we going to get a protein from DNA? In the ribosome, we talk about protein synthesis. Know about the genetic code, okay? How to interpret the genetic code, how to produce uh, a protein from the genetic code, okay? And also how to produce a protein from mRNA, okay? Then here there's protein purine synthesis and breakdown. This is now nucleotide metabolism. Okay, there are two purines. These are double ringed, right? So there's adenine and guanine. These are simply purines. So know about their synthesis and also the breakdown. The breakdown is important because we talk about accumulation of um, purines, and when there's excessive breakdown of them, where there's uh, uric acid accumulation and can settle in the joints and uh, cause arthritis, right? Pyrimidine synthesis as well, and mechanism of action of allopurinol in the treatment of gout. And what is gout as well? Okay. The Lesch-Nyhan disease, cancer and its molecular basis. How does cancer come about and what is cancer? And know about those genes when they are defective, right? The P53 gene, RB gene, okay? And those four molecular bases of cancer, okay? We talk about the growth promoting oncogenes, okay? When they have been activated to oncogenes, eh? Then we talk about the tumor suppressor genes, which I've mentioned. We talk about alteration in genes controlling apoptosis. Okay. We talk about alterations, changes in the DNA repairing machinery, right? Genes. Okay. Well, hormones as well. What are we supposed to know about hormones? Definition of hormones. What are hormones? And the classes of hormones. We've got um, the peptide types of hormones, right? Amino acid derived hormones. Uh, and also mm, the cholesterol derived hormones. These are lipid derived hormones, right? Yeah. Know the differences about them, as well as the release and the the action. How do they act in that case, right? These other hormones, we find that they're going to act through the extracellular receptors, through the second messengers. We talk about CAMP, okay? Cyclic adenosine monophosphate pathway. You explain all those things, right? The lipid ones find that they can go directly to the inside. They're able to cross the cell membrane. Okay. Vitamins and types of vitamins. ADEC, vitamin A, vitamin D, vitamin E, vitamin K, okay, and also regulation of gene expression and blood as well. Okay, so this document has been prepared and compiled by Dr. Rostod Max Banda from Rostod Academy. Thank you so much for watching.